So, a very good afternoon, good evening, everyone. We'll just wait for 2 3 minutes, uh, and then we'll start the class. Okay, so good evening, everybody. So, today we'll be covering the questions which were discussed, the topics which was taught in week 9 lecture. So, up till now, we have learned a lot about developmental biology. We started with the basics of what is developmental biology. Then we studied about the various kinds of alternative pathways, the expression pathways that are needed in developmental biology. The how the transcription, the expression of gene happens uh, in developmental biology. What are the alternate and the normal canonical mechanisms? We studied about the various developmental pathways that are involved, like Wind pathway. We have studied TGF beta pathway. We have studied about RTK signaling. Then we have also studied about the excess development in Drosophila in very much detail, Drosophila excess development, and other major concepts. But what all we have studied up till now was majorly related to the animal side, that is the animal developmental biology. So today we will be discussing the questions which are related to plant developmental biology. So week 9 majorly covers the portion of from week 9 the portion of plant developmental biology majorly starts. So today we will be discussing questions uh, related to plant developmental biology. So I hope you all have gone through the online recorded lectures so that it will be easier for you to solve the questions and to understand the concepts. Anyways, we are going to discuss the concepts or the topics which are related to the questions that we are going to solve today. So, we'll come to the first question. So, the first question is, so we'll do as we always do in a class. First, I'll give the question. I'll wait for a minute or two for you to solve the question. And then I'll switch to the screen and then we'll try to discuss it and understand the concept. Okay, so the first question is, which of the following statements regarding megasporogenesis are true? Um, Just a minute. Let me label it for you. A, B, C, and D. So A is the product of meiosis further undergoes mitotic cell divisions. B, unequal meiotic division gives rise to the megaspore. C, in megasporogenesis, the meiotic products directly form the embryo sac. And D, all four meiotic daughter cells divide once to form the eight-celled embryo sac. This is your question, and the question is that we have to tell that which all statements which are related to megasporogenesis are true. So there can be more than one correct answer. So I'll give you 2 minutes and then I'll switch the screen and then we'll discuss it. Okay. 1 more minute left. Okay, so let us discuss this question. Let me see. Okay. So the question is, which of the following statements regarding megasporogenesis are true? So first of all, what is megasporogenesis? So megasporogenesis, which is seen in megasporogenesis, is observed in angiosperms. Okay. So megasporogenesis is a process where a diploid megaspore mother cell, which is known as MMC, MC, which is megaspore mother cell, a diploid megaspore mother cell in the ovule undergoes meiosis and produces four haploid megaspores. This is the uh, process of megasporogenesis. So what happens, that megasporogenesis, it occurs in ovule. Okay, first thing is that it occurs in ovule and it produces four haploid megaspores. Four four haploid megaspores are produced, and from these four haploid megaspores, three degenerate and one remains functional. Okay, so there are total four haploid megaspores from which three, they degenerate, and one only one of the haploid megaspore, it remains functional. Then this one functional haploid megaspore, this undergoes three rounds of mitotic division. Okay, this undergoes three rounds of mitotic division. And then what happens? It forms a 8 nucleate embryo sac. It forms 8 nucleate embryo sac, which is the female gametophyte. Okay. Now we'll check the statements. First is, the product of meiosis further undergoes mitotic cell divisions. It is correct. Yes, it is correct, because the functional megaspore, the product of meiosis. So basically, what happens, that your diploid megaspore mother cell, it undergoes meiosis and it has formed four megaspores. Now, from these four megaspores, three are degenerate. The one which remains functional, it undergoes mitotic division. So that's what we have discussed. It undergoes three rounds of mitotic division and then it forms embryo sac. So this is correct. The product of meiosis further undergoes mitotic divisions. Yes, it undergoes how many rounds? Three rounds of mitotic divisions. Then unequal meiotic division gives rise to the megaspore. Unequal meiotic division gives rise to megaspore. True, because although meiosis produces four megaspores, only one survives. That is, there is only one functional megaspore and the other three are degenerate. So this is often described as an unequal in outcome. Okay, not in cytokinesis mechanism, but in fate it is an unequal outcome. So unequal meiotic division gives rise to the megaspore. True. Then in megasporogenesis, the meiotic products directly form the embryo sac. It is true or false? It is false. Why? Because the meiotic product, the functional megaspore, must undergo mitosis first to form the embryo sac. It cannot form the embryo sac directly. Then all four meiotic daughter cells divide once to form the 8-celled embryo sac. It is also false because only one megaspore survives. So not all the four meiotic cells are dividing, or three are degenerates, only one divide to form the 8-cell embryo sac. So 1, 2, and 3, 1, 2, and 4 are correct, and third is wrong. Oh, sorry, this is also wrong because only one is dividing. Okay, fine. Next question is, choose the correct statements regarding microsporogenesis. So up till now we discussed about megasporogenesis. Now the question is about microsporogenesis. All four, let me just Yes. So all four microspores generated from meiotic divisions can form the pollen. B. Microspore nuclei undergo two mitotic divisions to form a four-celled pollen. C. Each pollen contains two sperm cells capable of fertilization. And D. No asymmetric division takes place during microsporogenesis. Okay, so these are your four options. So up till now we discussed about megasporogenesis. Now the question is about microsporogenesis. Think and then answer. Think. Also think what is the difference between microsporogenesis and megasporogenesis. One minute more. Okay. So question is choose the correct statements regarding microsporogenesis. So first of all, what is microsporogenesis? Is a process in which a diploid mother microspore mother cell undergoes meiosis and produces a tetrad of four haploid microspores. So basically microsporogenesis it happens in anther. So megasporogenesis is a process where a megaspore mother cell in the ovule and here the megaspore mother cell in the anther. So it's a process in which a diploid microspore mother cell, in short we call as MMC, undergoes meiosis and it produces a tetrad of four haploid microspores. Okay, so there is a tetrad of four haploid microspores. Okay, then what happens in megasporogenesis? What happens that there are out of the four megaspores produced, three were degenerate and only one remains functional, but here all the four are functional. Okay, and each can develop into a pollen grain. There three were degenerate, only one can undergo further meiotic divisions to form the female gametophyte, but here all the four microspores are functional, and each of the microspores can develop to form a pollen grain. So each microspore undergoes two rounds of mitotic divisions. There were three rounds in megasporogenesis. In microsporogenesis, there are two rounds of mitotic divisions. Okay, fine. So just a minute, let me just see. Okay, so there are two rounds of mitotic divisions. So first mitosis is asymmetric, which forms a large vegetative cell plus a small generative cell. Okay, so the first round of mitotic division, it is asymmetric. Okay, which produces a large vegetative cell and a small generative cell. And the second round of mitosis division, basically it produces two sperm cells. The second round of mitotic division, it produces two sperm cells. So I hope this is clear. So basically, mature pollen, it has one vegetative nucleus plus two sperm nuclei. So there are total three cells. One vegetative nucleus plus two sperm nuclei. Okay. Now let us check the statements. First is, all four microspores generated from meiotic divisions can form the pollen. True, we have just discussed. All the four survives and all the four have the potential to develop into pollen. So first statement is correct. Then microspore nuclei undergo two mitotic divisions to form a four-celled pollen. Is it true or false? It is false. Why? Because the final pollen is three-celled. One vegetative nucleus is there and two sperm nucleus is there, not four. So the final nuclei is a three-celled nuclei, not a four-celled. One vegetative and two sperm. Then each pollen contains two sperm cells capable of fertilization. Yes, there are three nucleus, one is vegetative and two sperm. So each pollen has two sperms. Yes, this is also correct. And the fourth statement is, no asymmetric division takes place during microsporogenesis. Is it true or false? No, it is a false statement. Why? Because just now we have discussed that the first mitotic division is asymmetric, which generates a small generative cell and a large vegetative cell, and the second mitotic division, it produces two sperm cells. So this asymmetric division, it produces a small generative cell plus a large vegetative cell. Okay, so I hope this is clear. Now let us come to the next question, which is, choose the correct option regarding fertilization in plants. Just a minute, let me see. Okay, yeah, sorry for the interruption. So the question is, choose the correct option regarding fertilization in plants. Let me just label it as A, B, C, D, and E. So A is, perfect flowers cannot undertake self fertilization. B, fertilization is dispensable for embryo formation. C, POP2 needs to be expressed in both the style and the pollen for pollen tube formation. D, in angiosperms, double fertilization results in the formation of a triploid endosperm. And E, angiosperm endosperm is triploid. Okay, so you have 2 minutes. Try to answer this question. Think what can be the answer. You should know what is self fertilization and what are perfect flowers. 1 more minute left. Okay. So let us discuss this question then. Okay. The question says choose the correct option regarding fertilization in plants. So, uh, first option is perfect flowers under cannot undergo self fertilization. Is it true or false? Tell me. So, what is a perfect flower? Can anybody tell me what is a perfect flower? So, our perfect flower is something that cannot under that has a flower that has both stamens, that is male, and carpels, that is female part in the same flower. Okay, for example, pea, wheat, rice, they are perfect flowers and they can self fertilize. It can be, though self fertilization can be prevented only in certain conditions. For example, dichogamy. What is dichogamy? In which male and female parts mature at different timings. So there are different timings of maturation of male and female parts, and because the timing of maturation is different, they cannot fertilize. So that is known as dichogamy. And then hercogamy. Hercogamy means there's a spatial separation of male and female parts. So when somebody so the definition of a perfect flower is in which, sorry, female, which is carpel, and male part, which we call as stamens, they are present in the same flower. Okay, So now let us come to the options. First is perfect flower cannot undergo self fertilization. It is true or false? It is false, because just now we have studied that perfect flower can undergo self fertilization only in certain conditions like dichogamy, hercogamy, and some sexual incompatibility. For example, it cannot undergo fertilization. Then fertilization is dispensable for embryo formation. Yes, it is true, because see, in some cases embryos can form without fertilization also. So fertilization is not indispensable, it is dispensable. Then pop2 needs to be expressed in both the style and the pollen for pollen tube formation. Is it true or false? Yes, it is true, because pop2 encodes GABA transaminase which regulates the GABA levels. So both maternal tissue, that is style, and pollen, it requires proper pop2 activity for correct pollen tube growth and guidance. So this is a correct statement. You understood? So basically, POP2 encodes GABA transaminase, and both style and polar, it requires POP2 activity for correct pollen tube growth and guidance. So POP2 needs to be expressed in both style and pollen. Then in angiosperms, double fertilization results in the formation of a triploid endosperm. Yes, it is true. It is correct. So classical definition of this is that one sperm plus egg, so there will be a zygote. So basically triploid endosperm means one sperm plus two polar nuclei. Okay, that will form a triploid endosperm. One sperm. Okay, then angiosperm endosperm is triploid. Angiosperm endosperm is triploid. It is true or false? It is true. So standard case triploid is means 3n. Exceptions, there can be some higher ploidy endosperms, but normally angiosperm endosperm is triploid only. So is it correct? Just go through the options to see whether you could understand or not. Okay. So let us move to the next question. So these are the accepted answers. Fertilization is dispensable for embryo formation. POP2 needs to be expressed in both the style and the pollen for pollen tube formation. In angiosperms, double fertilization results in the formation of a triploid endosperm. And angiosperm endosperm is triploid. Okay, just a minute. Okay, next question is select the options with the correct chronological order of the formation of structures during plant embryogenesis. Okay. So A is basal cell, two cell embryo, hypophysis, shoot apical meristem. Or B, two cell embryo, then basal cell, then hypophysis, then shoot apical meristem. C, hypophysis, then basal cell, then two cell embryo, then shoot apical meristem. D, shoot apical meristem, then basal cell, two cell embryo, hypophysis. Or E, basal cell, two cell embryo, hypophysis, or cotyledons. So you have to see the correct chronological order of the formation of structures during plant embryogenesis. Pick the correct chronological order. Okay, I'll give you 2 minutes, then I'll switch the screen. Try to answer. One more minute left. 30 more seconds. Okay, let us discuss the question then. Okay. So we have to select the correct chronological order of the formation of structures during plant embryogenesis. Okay. So see, I'll tell you the correct chronological order. So first what happens, that zygote is there. Okay. It divides asymmetrically and it gives rise to a basal cell and an apical cell. Okay. So it gives rise to a basal cell which is towards the micropyle which forms the suspensor, and then there is a apical cell. So we have to start with basal cells options. There are two basal cell options. Okay. Then next what happens, that there is a two cell embryo state. Then there is a two cell embryo state, that is two cell embryo which is like apical and basal. So both have two cell embryo state. Okay. Then what happens? Then basal lineage produces the hypophysis, which is the uppermost suspensor cell. And this hypophysis later contributes to the root apical meristem plus root cap. That means next, what is the uh, organ which is getting formed? Which this is, it is hypophysis. Hypophysis. So this hypophysis is the uppermost suspensor cell. And then what happens? After hypophysis, now comes the difference. Up till here, these two were literally the same. Then the only difference is shoot apical meristem or cotyledons. But the catch here is that hypophysis can give rise to both shoot apical meristem or cotyledons. Okay. So during the globular stages or during the heart stages, shoot apical meristem and cotyledons both can be formed. So this means, I'll just write here SAM, which is shoot apical meristem, SAM or cotyledons. Both can be formed. So this means that A is also correct and E is also correct. Okay, so there are two correct options for this question. Basal cell, two cell embryo, hypophysis, shoot apical meristem. Basal cell, two cell embryo, hypophysis, and cotyledons. Next. Refer to the following table and choose the option which with all correctly matched pairs. So the question is, refer to the following table and choose the options with all correctly matched pairs. A. Microsporangium. B. Megasporangium. C. Microspore. D. Triple nuclear fusion. E. Embryo sac. And on the other side, diploid, haploid, pollen, endosperm, and synergids. Okay, so I'll give you 2 minutes and then we'll discuss the question. So there can be more than one correct answers in this case, okay? Because microsporangium can be related to more than one options which are given on the right side. You have to choose accordingly. Okay, let us discuss the question then. Just 1 minute. Hmm. So refer to the following table and choose the options with all correctly matched pairs. Okay. So first is microsporangium. So microsporangium is basically a diploid sporophytic structure which is anther locule. So it can match with diploid. That is, this can be the this can be option A1. Then megasporangium. It is a diploid sporophytic structure and it can match with deployed, but we cannot match it because of the ambiguity, so let us just remove it. So we have not, that is why in this option you can see B is not there. Then microspores, these are haploid products of meiosis which develop into pollen, so this can be matched to haploid, so C2. Then D is triple fusion nucleus produces endosperm, so D can be 4, but here it is not given, but that is okay. Then embryo sac, female gametophyte that contains the synergids and egg polar nuclei, so it can match to synergids. So one of the answer that looks is here is this one. Okay. Then can there be more than one options? Yes. See, A can match with 3 as well. That is microsporangium pollen, because it microsporangium and microspores are associated with pollen, because so therefore this can also be correct. Then C can match with third, that is microspores pollen again, because microspores has pollen in it. Then E can match with fifth, which is embryo sacs, because which is redundant, but this is also true. So this can also embryo sacs uh, can match with synergist, same as we have done it before, because female gametophytes contain synergist. So this can also be a correct option. Then can are there any more? I think this can also be correct. See, B matches with 5. Megasporangium, synergists, yes, because megasporangium and endosac are similarly. Then your C is third. Microspores, pollen, yes, and E 5 to hai. So there can be more than one correct option. So these are the all the correct options that we can have here. Okay. Now let us come to the next question. This is a pretty nice question. Students experimented to study the time the pollen tube took to reach the egg chamber. Just 1 minute. Okay. Hmm. Question is, students experimented to study the time the pollen tube took to reach the egg chamber. RFP tagged pollen 1 and GFP tagged, which is pollen 2, pollens were transferred to the stigmas of the same plant from which these pollens were taken. The following graph depicts the result obtained. Based on this information, select the correct option regarding the phenomena involved. Okay, your options are 1, 2, 3, 4, 5, 6. First, why there was this sound? Anybody? No. Okay. So, hmm. First option is pollen 1 genotype may be S1 and the stigma is S1 oblique S3. Pollen 1 genotype may be S2 and the stigma is S1 S3. Pollen 2 genotype may be S1 and the stigma is S1 S3. Pollen 2 genotype may be S2 and the stigma is S1 S3. Pollen 1 genotype may be S1 and the stigma is S2 S3. Or pollen 2 genotype may be S2 and the stigma is S2 S3. Okay, these are your options. So I'll give you 3 minutes to answer this question, and then I'll switch the screen, we'll discuss it. 2 minutes more. 1 minute more. One more minute left. Okay. Let us discuss the question then. So I think the answer was already, I've opened it anyways. So the question is that students experimented to study the time the pollen tube took to reach the egg chamber. RFP tagged and GFP tagged pollens were transferred to the stigmas of the same plant from which these pollens were taken. The following graphs depicts the result obtained. Based on this information, select the correct option regarding the phenomena involved. Hmm. So basically, this phenomena, the phenomena that is dealt in this question is S locus self incompatibility. Okay, it is S locus self incompatibility. So what does it mean? This phenomena is it's a genetic mechanism in flowering plants that prevents self fertilization and promotes cross pollination. So this phenomena, it prevents self fertilization and it promotes cross pollination. Basically, what happens? It is controlled by a highly polymorphic S locus. The S locus encodes protein which is expressed in pollen and in the stigma style that interact to determine the compatibility. So, what happens is pollen is rejected when its S allele matches an allele in the stigma. Okay, this is important. I am writing it over here that pollen is rejected. Note that there is rejection of pollen and not of the ovules. Pollen is rejected when its when its S allele matches and allele in the stigma. Okay, so now let us look at the question. Here we can see that pollen 1, which is red in color, it grows. Whereas pollen 2, which is green in color, it fails to grow. So if we apply this gametophytic self-incompatibility logic, a stigma heterozygous for two S alleles will reject any pollen whose haploid S matches either stigma allele. Okay. So a pollen grain is accepted only if its S allele does not match either allele which is present in the stigma. So if it is S1 and S2 and stigma is also S1 and S2, then it cannot happen. So from the graph, we can see that pollen 1 is compatible. That means it is growing, and pollen 2 is incompatible. So the stigma must carry an S allele that matches pollen 2 but not pollen 1. Because that is why the pollen 1 is unable is able to grow and pollen 2 is unable to grow. Now here you see pollen 1 genotype may be S and the stigma is S1, S3. Yes. See, pollen 1, is it correct? No, because here they are matching. So this is not correct. Pollen 1 genotype may be S2 and the stigma is S1, S3. Yes, because pollen 1 is growing. So you see this is S2 and it is not matching with the stigma. So this can be correct. Pollen 2 genotype may be S1 and the stigma is S1, S3. It can be correct because there is S1, S1 is matching. Therefore it is incompatible, so it is unable to grow. Pollen 2 may be S2 and the stigma is S2 and S3. No, it cannot be done because if it is the case, it should be compatible and it should grow, but it is not growing, so this cannot be the case. Pollen 1 genotype may be S1 and the stigma is S2, S3. This can be the case because the genotype of the pollen is not matching with the stigma. That means it is compatible, therefore it can grow. See, the genotype of the pollen should be different of that of stigma so that it can be compatible. Pollen 2 genotype may be S2 and the stigma can be S2, S3. Yes, this is also correct. So pollen is rejected when its S allele matches with the allele in the stigma. For example, here you see the S allele of pollen is S2 and the stigma also has S2. So this pollen is rejected in that case. Therefore, it becomes incompatible. So that is the reason why here you can say that the allele of pollen, S allele of pollen, has matched with the allele of the stigma, and that is why it was unable to grow. But here it was different, so it become compatible, it grew. Okay. Now next question is, a bacterial protein X is shown to induce SRK activation in a certain Brassica species. Based on the given information, select the option that describes the effect of protein X in this particular Brassica species. Okay, so these are the options. Let me just label it as. So A, the stigma of plants infected with the bacteria will successfully undergo self-fertilization. B, the stigma of plants infected with the bacteria will successfully undergo cross-fertilization. C, expression of protein X in stigma will inhibit pollen tube growth upon self-fertilization. And D, expression of protein X in the pollen will inhibit pollen tube growth upon cross-fertilization. So these are your four options. Think what can be the answer. One more minute left. Okay, let us discuss the question then. So, the question says a bacterial protein X is shown to induce SRK activation in a certain Brassica species. Based on the given information, select the option that describes the effect of protein X. So, basically, SRK is a receptor kinase on the female surface that is activated by a pollen borne ligand, which is SCR or SP11, which is present at the pollen surface. Activation of SRK, it triggers the self-incompatibility signaling cascade in the stigma and it inhibits pollen tube growth. Okay, so what does it says? It says that activation of SRK, if SRK is activated, then what happens? It triggers the self incompatibility signaling cascade. Ultimately, what happens? So, self incompatibility signaling is triggered and it inhibits pollen tube growth. Okay, so for SRK to be activated during pollination, a ligand must be present on the pollen surface. Okay, or presented to the stigmatic receptor at the surface. So now according to this is first is the stigma of plants infected with the bacteria will successfully undergo self pollination. Is it true or false? It is false, because if the bacterial protein X activates SRK activation triggers signaling this SI pathway. Okay, and prevent self pollen from fertilizing. So, infection that activates SRK would block, not enable selfing. So, this is wrong. Then, second is the stigma of plants infected with the bacteria will successfully undergo cross fertilization. It is also false for the same reason that activation of SRK at the stigma it prevents pollen tube growth. Thus, cross pollen would be blocked if SRK is activated. Then, expression of protein X in stigma will inhibit pollen tube growth upon self fertilization. Again, it is not accepted because mere expression of X inside stigma tissue does not necessarily present an SRK activating ligand at the extracellular pollen stigma surface. SRK activation, it requires ligand presentation to the receptor on the stigma surface. So, expressing X in the stigma itself may not mimic the pollen borne activation in the context of pollination. The accepted interpretation is that pollen borne expression or presentation of X is what causes SRK activation during pollination. Okay, so now comes the fourth. Fourth is expression of protein X in the pollen will inhibit pollen tube growth upon cross fertilization. Is it true? Yes, it is true. Because if protein X behaves like the pollen ligand or otherwise can prevent the activating signal at the pollen surface, pollen that expresses X will activate SRK on the stigma when it lands there. That activation triggers the SI response and blocks pollen tube growth, even during what would normally be a cross. So this is consistent with the accepted result. So option 4 is the correct answer. Then now comes the next question. An S1 S2 plant is crossed with an S2 S3 plant. Assuming only gametophytic self incompatibility, what is the percentage of progeny with S1S2 genotype? Okay, I'll give you 3 minutes to answer this question and then I'll switch the screen. Just a minute, I have just one thing to see. Uh, okay, fine. Then. Hmm. I'll give you 3 minutes. Try to solve this. 2 more minutes. One more minute left. Okay. Now let us solve this question. See how you have to do this question. So parent 1 has the genotype of S1, S2. Parent 2 has the genotype of S2, S3. So, gametes, if there is no incompatibility, what will be the possible pollen? S1, S2, S2, S3. So this would be S1, S2, S1, S3, S2, S2, and S2, S3. Okay. These could have been the possible pollen without any incompatibility concept. But now we have to filter the compatibility thing. So what happens when we apply gametophytic self-incompatibility concept? Because here they are asking about gametophytic self-incompatibility concept. So what happens? The rule is pollen is rejected if its haploid S allele matches either allele of the stigma. Okay, so pollen here has S1 and S2 and stigma has S2 and S3. That means S2 pollen is rejected. Remember, we have to reject the pollen and not the egg. Rejection is pollen specific and not egg specific. Let me just write here. Rejection is pollen specific and not egg specific. So here rule is pollen is rejected if its haploid S allele matches either allele of the stigma. So these are the two alleles of the stigma, S2 and S3, and these are the alleles of the pollen, S1 and S2. So here this S2 allele of the pollen, it matches with the S2 allele of the stigma. So S2 allele is rejected. So now the combinations can be S1 S2 or S1 S3. So 50-50% chances there. So the answer is 50%. Each has equal probability. So progeny distribution would be 50%. Now you can say that how can we have S1, S2? Because gamete is not rejected. Pollen is rejected. S2 pollen is rejected. Now this S1 pollen, it can match with, it can form with S2, it can form with S3. S1, S2, these are two different. No. So that is why this pollen has been rejected. So therefore the answer is 50%. So I hope this concept is clear. The rule is that pollen is rejected if its haploid S allele matches either allele of the stigma. Okay. So this is your GSI, that is gametophytic self-incompatibility. So the rule is a haploid pollen carrying the S allele is rejected if the female parent pistils contain this is the same. What I have told you, that is the thing. Next question is, an S1 S2 plant is crossed with an S2 S3 plant. Assuming only sporophytic self incompatibility, what is the percentage of progeny with S1 S2 genotype? Now, here comes sporophytic self incompatibility. So, what happens in sporophytic self incompatibility? In sporophytic self incompatibility, the pollen phenotype is determined by the diploid genotype of the pollen producing parents or the pollen producing plant rather than by the pollen's own haploid genotype. Okay, here in your gametophytic self incompatibility, what happens? That the pollen phenotype is determined by the pollen's own haploid genotype. But here, the in sporophytic self incompatibility, the pollen phenotype is determined by the diploid genotype of the pollen producing parent and not by the pollen's own haploid genotype. This is the difference. So what happens? Pollen, so here what would be the answer? The answer would be 0. Why? Because pollen from an S1 S2 plant presents both S1 and S2 and will be rejected by the stigma carrying S2. Hence, no fertilization produces S1 S2 progeny. So I hope this is clear that here the pollen phenotype is determined by pollen producing plant. Okay, plant rather than by the pollen's own haploid genotype. This is important. Okay. Next and the last question for the day is green fluorescent proteins. Green fluorescent protein is expressed under auxin inducible promoter in cultured plant cells. Under which of the following conditions will GFP not be expressed upon auxin treatment? So let me just mark it as A, B, C, D, and E. A. Loss of function mutation in OXIAA only. B. Loss of function mutation in ARF only. C. Loss of function mutation in 26S proteasome. D. Administration of anti-ubiquitin antibodies. Or E. Loss of function mutation in SCF. Okay, so you have uh, 2 minutes. After 2 minutes, I'll switch the screen and then we'll discuss it. Okay. One more minute left. Okay. So let us try to understand this question. So first of all, let's have some basics of auxin signaling. So when auxin is present, it promotes ubiquitination and degradation of the AuxIAA repressor. So when auxin is present, then what happens? Then there is ubiquitination and degradation of AuxIAA repressor. Okay, and this degradation of AuxIAA repressor is mediated by SCF-TIR1 ubiquitinase ligase complex. And once this AuxIAA repressor is degraded, then ARFs, which are known as auxin response factor. Okay, auxin This AIRF are free to activate the auxin inducible gene. For example, here we can say as GFP in this case. So now here the question is, under which of the following conditions GFP is not going to be expressed? Not expressed. This is important. So A, loss of function mutation in AuxIAA only. Is it correct? No. Why? Because AuxIAA are repressors. If they are mutated and cannot repress, ARFs will activate transcription even without auxin. So GFP will still be expressed, maybe even constitutively. So this is wrong. Then loss of function mutation in ARF only. ARFs are the transcription factors. Without them, no matter what happens to the AuxIAA, transcription cannot be activated. So yes, it is correct. In this case, GFP will not be expressed. Loss of function mutation in the 26X proteasome. Again, it is correct. Why? Because AuxIA cannot be degraded if proteasome is only losing its function. So they remain bound to the ARFs, transcription stays off. So GFP will not be expressed. Then administration of anti-ubiquitin antibodies. Yes, ubiquitination of AuxIA is blocked, AuxIA is not degraded, and repression persists. So GFP will not be expressed in this case also. Then loss of function mutation in SCF. SCF complex is required to ubiquitinate AuxIAA. So without it, AuxIAA cannot degrade, so repression will persist. So in this case also GFP will not be expressed. So this is also correct. Okay. So I hope this is clear to you. Yeah. So important thing just I wanted to show here. Always remember that auxin, if it is present, then ultimately there will be transcription of the auxin inducible gene through ARFs, which are nothing but auxin response factor. And if auxin is not there, then what happens? This AuxIAA is a repressor. This will block the transcription. Okay. So transcription would be blocked in this case. So this is the main, like, the concept behind it. So I hope this is clear to you. With this, we have completed all the questions of week 9. So week 9 was related to plant developmental biology. If you have any questions, any queries, you can always ask me. You can always comment in the YouTube videos that we upload every week for these lectures. You can ask there also, or you can ask in the classes also. So I hope you were able to understand, and we'll meet in next week with week 10.